about your this week assignments that is mainly focused on your different aspects of muscles and muscle contraction, and we are going to have a view on that. And before going to the theory part, practical question answers, I'll go a bit concept related to that. And if you find anything difficult, you can just raise your hand, or you can just unmute yourself and go ahead. And okay, so let's start with the first slide that I have for you guys. So this slide is much familiar with you, right? So this is basically a introduction to different types of muscles. Okay, now what are the different muscle types, and on what basis the configuration of a muscle decides or depicts the functionality of a particular organ? Okay, so if you find the first type of muscle is your skeletal muscle, right? Skeletal muscle is a muscle that is mainly one of the strongest and strongest muscle that is having striation. Now striation is the line found in between the muscles that provide strength, energy, and it basically possesses mitochondria so that it has more strength as compared to the smooth muscles. Okay, so this is basically, since it is the one of the mechanical muscle in our body that performs measure of the work, it is responsible, and it comprises the part of the muscular organs like your hand, legs, etc. Okay, the second type of muscle that we have is your smooth muscle. Okay, now the smooth muscles are the muscles that mainly comprises the structurality of your vital organs. Okay, now what are the vital organs? So that is basically comprises the inner lining of the body, that is majority of the organs that are results of the mesodermal origin. So this is basically comprises of smooth muscles, and these muscles are nothing but these are very simple type of muscles having a single nuclei, and they don't have any striations. Okay, rather they don't need to be mechanically stable or mechanically strengthened, but they need to be vital, as they are very comprises of very important organs in our body, and that they are protected by our skeletal muscle externally. Okay, so basically all the vital organs are comprised by smooth muscles, and the special category of muscle is your cardiac muscles. Okay, now cardiac muscles, if you consider it, it is comprises of both skeletal and smooth muscles. Now why it is so? If you find the cardiac muscles is mainly found in your heart only. Okay, now heart is one of the busiest organ in your body that works for 24 into 7. Okay, now if you see how it is mechanically activated, it pumps throughout the body 24 into 7, right? If it stops for even a minute or even 3 4 5 minutes, it leads to much more complexion that even we can expect. Okay, so it needs to pump throughout the day 24 into 7, so that for that purpose it needs a lots of energy, and basically the energy is provided by the energy currency that is your mitochondria. Okay, I would like to ask you guys one question. Are you aware of the hummingbird? Okay, so if I'll ask you that if you find that hummingbird flies wings very much fast, that is 60-70 times in a minute. Okay, now if I'll ask what are the compositions, or what are the criteria, or what are the characters its muscle possesses that is responsible for the such strongest behavior of that particular organism? Yes, myoglobulin, as you are mentioning, is one of the reason, but the point is that myoglobulin is the molecule that provide oxygen. Okay, and muscle needs energy to work. Okay, that oxygen get converted to energy by means of a process called your respiration. Okay, and for respiration you needs mitochondria. True. So more the amount of mitochondria, more the amount of energy will be produced, and more the amount of the strength or the functionality of the muscle will increase. So here I can say that possesses lots of amount of mitochondria that helps in the process of that fastening movement of that particular organism. Now if you consider the cardiac muscle, you'll find it has a rhythmicity of it pumping throughout the day. Apart from this, it is a part of your vital organ. Okay, so it needs to be protected, and it's need to be strength one of the strength muscle as well. And apart from these, there are so many plexus, there are so many muscles, or that helps in the process of regulation of the heart. That's why it comprises of your both skeletal muscle, smooth muscle, along with, you can find here, you can see there is a junction in between two layer of the muscles. Okay, so this slide is representing basically your different type of muscle, that is your smooth muscle, skeletal muscle, and your cardiac muscle. Clear? Okay. Now if you find this slide, actually we are facing a bit network. So here you can see in orange it is calcium molecule that is basically binds to that blue color troponin that is responsible for the activation of myosin binding site of that particular actin molecule, and that is responsible for the movement of your muscle. That is basically leads to the process called muscle contraction. Okay, so basically I have put one video here, but due to some network issue I'm not being able to play it there, but I have given the link in your PPT, so you can just go into that to understand and find how muscle basically contracts. Okay, so it's not that complicated, rather it's just a binding of calcium molecule to the troponin that activates the binding site of the myosin head, and that's basically responsible for the muscle contraction. Okay, now if you consider the structure here given, the question is that, now let's come to the first one, that is, what is the membrane covering muscle fiber, or okay, what is the membrane basically that covers your muscle fiber? Now if you find here, then see, there are several bundles that are comprises, and individual bundles are called your myofibrils. Okay, now if you find there are so many myofibrils comprises to form one uh, muscular bundle, that is basically your uh, that is basically comprises your muscle layer. Okay, now this is covered by one membrane that is called your sarcolemma. Now why lemma? Whenever you will find in biology, the lemma what represents the membrane, and sarco is basically to depict your muscle there. Okay, now if you'll find the specificity of sarcolemma is that if you find sarcoplasmic reticulum there, you will find different ions that is released from that sarcoplasmic reticulum and is responsible for your contraction of the muscles. Now if you find one myofibril comprises of several sarcomere. Okay, now sarcomere is the structure that is individual units of the myofibrils, and that is responsible, and that is basically comprises of your actin and myosin that contracts in order to results in your muscle contraction and relaxation. Okay. Okay, now if you find here, see, you can find here that if, as I mentioned, the myofibrils further divided into very smaller subunits that are called your sarcomere. Okay, now if you see, there are so many sarcomeres present in one myofibrils. Now let's understand the structure of the sarcomere in details. Okay, that will make you clarifiers to understand the muscle contraction. Okay, so see here, if you'll find I have presented one unit of sarcomere. Okay, that is basically comprises of different lines. Okay, now here it is important to understand the individual lines. Okay, so you'll find in green that is your Z line. Okay, that Z line basically represents the boundaries of individual sarcomere. Okay, so one sarcomere comprises of two Z line. Okay, now the Z line is present in between your I band. Okay, as it is mentioned here. Now I band, what I band is that? I band is mainly comprises only of thin filaments. Okay, what are the thin filaments then? Thin filaments are only comprises of actin molecules, that is basically G actin as well as your F actin. Basically smaller unit of G actin comprises to form your fibrous actin, that is your long fibrillary actin, and that basically comprises of your I band. So it is the reason I band is the reason that is not overlapped actin molecules. Okay, now if you consider the reason in between, then that is your myosin, and myosin, the entire myosin molecule calls the region that is comprised by the entire myosin molecules is called your A band. Okay, now the myosin have it has a head and it has a tail region. Okay, that myosin head is bind to the binding portion of your actin molecules in order to perform muscle contraction. Now in order to understand that concept, here I have given you one question you can find, and we are going to solve rather several questions based on this particular diagram. Okay, so you'll find in myofibrils the bands that only contains actin filaments. Okay, as I have mentioned, the reason that is contain only actin filaments is your I band, that is basically light chain that comprises only light chain, that is not overlapped I band, not overlapped actin molecules comprises your I band. Okay, similarly the region comprises by only myosin molecules and some overlapped actin molecules is called your, if you'll find the next question will be that. Okay, so the region that is comprises of your myosin molecules in blue is basically your, as you can mention, it is mentioned here, that is your A band. However, the region that is not overlapped of that particular myosin molecule is called your H zone. Okay, so I'll just put forward one question that if the muscle is contracting, then what will happen to your this I band? Can anybody of you can just answer the question? The question is that what will happen to the I band if the muscle is contracting? Yes, true. So the muscle will get, the I band will get decreased. Okay, you can see because it is overlapping, it is overlapping, the much more region of the I band will overlap the myosin molecules, the region of the I band will gradually decrease. So now the question is that I can ask you one more question is that what will happen to the A band then? Okay, just you can go ahead, if you can just, you would like to answer, just go ahead. Now what will happen to the A band? As I have already mentioned, what is basically A band? A band is the region that comprises your myosin molecules along with your actin regions that overlapped it. Okay, now so if that is the point, then the A region will remain constant. Okay, why? Because see, the length of the myosin is not decreasing at all, and whether it is overlapped by the actin molecules or not overlapped, it is basically not a matter to consider whether the A band is increasing or decreasing. So the A band will remain always constant. Okay, now what will happen to the length of your H zone? That is going to basically decrease with increase in your muscle contraction, because you can see the more region of the myosin will overlap, so the H zone will decrease I band will decrease, however there will be no impact on the A band. Okay. Now the third question is that in myofibrils, the band that contain only myosin filament and overlapping end with the actin filament. I have already discussed several times. It will be your A band. Okay. Okay. So next question is that in myofibrils, which of this is an anisotropic to polarized light? The question. So you can just let's understand the question that in myofibrils, that is in muscle layers, which of the structure is your anisotropic to polarized light? Suppose you are putting polarized light, then which of the structure is anisotropic? And similarly, just antagonist to that, in myofibrils, which of this is your isotropic to polarized light? Okay. Now consider that uh, what is anisotropic then? If you'll find the myosin length, so it is not a uniform length throughout the molecule. Okay, so it is different length at different positions because it has myosin head, it has tail. So if you consider the A band that is mainly comprises of your myosin molecule is A band that is mainly comprises of my myosin molecules. Due to the difference in the structurality of the myosin molecule, the length will be different from different angles. Okay, just wait. I'm looking at it. I think it is visible from my side, but let's wait and see. Uh, so can anybody else confirm that whether now it is visible or not? Because I think it's fine to me. If the screen is visible, can just anybody go ahead and reply? Okay then, thank you. So we were discussing that what are the anisotropic structure and isotropic structure. So if you'll find the actin molecular region, that is mainly your I band, is comprises of linear actin molecules. Okay. Now the linear actin molecules have a uniform length throughout the molecule. Throughout the fibrillar actin, you will find a uniform length. So with respect to the polarized light, you will find a similar kind of appearance. Okay, so that's why it is called your isotropic band, or the structure is called isotropic. Okay. However, if you consider the myosin structure, due to the structural difference in different layers, like you will find it has myosin head, it has tail, it has heavy portion, it's a light portion. So the structural appearance with respect to the polarized light will be different at different positions for the myosin molecules, that leads to depict that your A band is anisotropic in nature. Okay, so this is basically a difference between your isotropic I band and anisotropic A band. That's why it is also called I band and A band. I stands for your isotropic band, A stands for your anisotropic. Okay, because you will not find a similar appearance because A band has different reasons. It has H zone, it has bind overlapped zones. Okay, however, I band is your light chain region, and you will find a constant isotropism in the appearance of actin molecules. Okay. Any doubt till now? If you have any doubt, just you can go ahead. Else we will go for your question number 6. Anybody? Okay, just a minute. Okay, pardon me. So we were in the discussion of your isotropic and anisotropic band. So it's, I think it is clear to all of you. Okay. Okay. So now going to the next question is that uh, yeah, so during relaxation you will find the structure will again get differentiated, and you will find your I band will increase and your A band will remain constant as it is. However, the HGN is also going to expand, or it will increase in your length. Okay. Okay. So see the next question is that if you find that now how these things occur, how this muscle contraction is taking place. Now, what is happening to this? Now, how we are able to understand this process to occur? Now, for that purpose, this question will much more helpful, that you will find an axon potential leads to the secretion of fluid in local area of muscle fiber in the twist regions. Okay. So if you are aware of the system, that reflex system, you'll find we have several reasons. Okay, so just let me make you aware of the reflex system. Is that suppose you uh, touch some object, a hot object. Okay, now your brain tells you to just put your hand away from that particular position. Okay, so in order to take your hand back, your brain is going to signalize your muscle at that particular position, and that is basically going to act as a presence of neuromuscular junction, and that is basically the new nerve endings that resides in a particular ends in the muscle layer is called your neuromuscular junction. So that signalize you to pull your hand back. Okay, so now your reflex system, that is your CNS, that is the central nervous system, will release some stimulus, release some signals to the neuromuscular junctions. So that is basically a signal to pull your hand back. Okay, so as from this portion you can find now that particular signal when released to your muscle will really results in the process of muscle contraction. Okay, so it's not like that. The muscle contraction is associated with the nerve signals that is basically released by your nervous system. Okay, and that is found in a region called your neuromuscular junction. Now if you find here there is a term called motor neurons. Now what is motor system then? So as I told you that when you will find some effect, that it was a hot body. Now your reflex put some effect, that is start from E, that just take your hand back from that particular object. Okay, now this effect and effect is controls by your effector system and your effector system. Now in order to process that effect, out of that effect your nerves or your reflexes signalize to a particular nervous system that is called your motor system. So that is basically going to results in by in associated in association with your signals from the reflexes from your CNS to perform some action. Okay, so that is basically the motor actions that needed to be taken place. If you consider here other example, like other options like acetylcholine, glutamine, these are also neurotransmitters, but you will find hippocampus is not the organ that is responsible for muscle contraction. However, glutamate is also one is a neuroinhibitor, but it is not a neurotransmitter. Okay, so basically in order to process any actions, it is your acetylcholine that is basically secreted into the neuromuscular junctions, as you can see in here in yellow bubbles, and that will be released to the sarcoplasmic reticulum, and from that you will find the release of your calcium ions that will bind to your troponin molecule in the muscle layer or in your actin molecules, So that will activate the regions of binding for myosin molecules of the actin, and that will result in the process of muscle contraction. Fine. Okay. So you'll find that it has several other things, like there are inhibitory neurotransmitters, there are acceleratory, see, you can see the neurotransmitter that accelerate one action or that remove one actions. So you can find different categories. You can just go and have a look at that, and we can discuss in several other time. We'll find some time, then we are going to discuss about different categories of neurotransmitters. However, you should have aware of all the things, and you can just go and have a look at that and come back to me. You will find any doubt regarding that. Okay. Okay, so I think everything is fine, right? My screen is visible to all of you. Okay. Now, see. Now we are going to discuss about different aspects of you can find here that there are different things like uh, what will happen in the neuromuscular junction is actually basically there were uh, different neurotransmitter, as I told you. Okay, so that reason is basically regulated, and that response, that stimulus results in the communication of transmission of signals from one action terminal to the other action terminal in a way that it transfers signals. Okay, and basically as a result of change in the ions, you must be aware of that release and intake of your sodium and potassium ions. Okay, so now the question regarding just like that which ion cause the forces among actin and myosin filaments, causing them to slide along each other. Okay, now as I have already mentioned several times, you will find there are sarcoplasmic reticulum, that is, or a sarcolemma, that is basically the membrane of a muscle layer, and that possesses sarcoplasmic reticulum, that is basically equivalent to the endoplasmic reticulum of the other parts of our body. Now that particular structure is responsible for releasing different ions, that is specifically calcium ions. Okay, now that calcium ion is responsible for binding to the troponin molecules of the actin fibrous actin molecules, and that responsible for the activation of the ATP or binding sites for the myosin head, and that is responsible for the process of muscle contraction. Okay, so in that way it binds to the basically, see, if there is action between that two, that is your actin and myosin molecules, so they must are binding, and that is basically because of that attraction between that two molecule, that is your actin and myosin molecule. Now if you find here this does not have similar effect on muscle fiber as acetylcholine. Okay, so see here you can find papain is your a enzyme that is basically have inhibitory function as compared to acetylcholine, because acetylcholine is a neurotransmitter. Okay, so basically it helps in transmission of signals, first signals, as fast as possible in the neuromuscular junction. Now you will much aware of the other like methylcholine, carbacholine, nicotine. These also have the ability to act as a neurotransmitter. They act a neuromuscular junction. You must be aware of nicotine, that you'll find taking nicotine will relaxes your nerve endings, and that is delayed the signal transmission. So that's why you'll find but still it's a neurotransmitter. Okay, so it is positively affecting your signaling from one end to other end. However, papain is your one of the inhibitory enzyme that delayed the signal and that does not help in the process of nervous transmission. Rather, it will uh, antagonistically act along against the acetylcholine in order to perform the function of your nerve transmission. Okay, so if it is that in the neuromuscular junction, it act as an inhibitory enzyme. Rather, papain is one of the enzyme that inhibits the nerve transmission in a better or positive manner. Okay, okay, so just you can is this block. Okay, it's just block the nerve transmission. Okay, now if you'll find that other neurotransmitter are like there are so many neurotransmitters. You'll find GABA, you will find glutamate. All these are helps in the process of nerve transmission. Okay, now they are divided into different classes that, as I told you previously, you should just go and come to me for discussion if you find any doubt. Okay, now the question number 9 is that the muscle fiber consists of myofibrils. Each myofibrils has how many actin and how many myosin molecule? Okay, so in order to understand this question, let's go back to this figure. You'll find here I have represented your actin molecules in red. Okay, so you'll find the structure. Suppose this is a myosin molecule, so the actin is present at both of the end. It is overlapped by your myosin is overlapped by your actin molecules. So if you will consider here, there are four actin molecules. Okay, there are four actin molecules. You can see start from one that you will find in red, that is your thin filaments, is basically your actin molecules, of which in between your gel line is present. Okay, now if you'll find that both of these, like there are, it requires four actin molecules to cover one myosin molecule. Similarly, if you're gonna add myosin molecules, you need to add two actin molecules. So it is occurs in a way that you need two actin molecules to add in order to add one myosin molecules. Okay, so that simply means that the number of actin molecules will be double as compared to your myosin molecules. Okay, so the next question is, your heat exposures above 50 degrees Celsius leads to the shortening of muscle during use and is called what? Okay, so if you'll find, okay, we'll discuss the question 9 again this time. I think some background noise is coming, right? Can you please all mute your mics? Okay. So if you will find here the structure here as it is showing to all of you, see the reasons in the red is your actin portion. Okay, and the reasons in the blue. Okay, somebody is raising his hand. You can just turn your mic and you can just speak, or you can just write it in the chat box. Okay, you just don't need to raise your hand. So if you'll find here the structure that is showing to all of you, just a minute. Actually, we are facing some network issues right now. But okay. Okay, so if you can see this figure, you'll find that the reason in the okay screen is, I think it is okay from my side. You can just see whether we take with your new network and all these things. Okay, so if you'll find the structure, just let me go to the question again. That the question number 9 is your that the muscle fiber consists of myofibrils. Each myofibril has almost how many actin and how many myosin molecules. Okay, now if you'll find the options, that here is only single option where the number of myosin molecule is more than your number of actin molecules, and as we have discussed several times, and you will find the structure rather, uh, I'll take to you guys to just a minute. Screen is still not visible. Okay, just let me go through it, okay, just hold on a minute. Okay, so I think now it is visible, right? Actually, we are having some network issues, so just pardon for the delay. I'm just sharing my screen. Just hold on, okay? Okay, I think now it's visible to all of you, right? Okay, so let's discuss the question number 9 that you have asked. So we have asked how many actin myosin molecules needed to cover one myosin molecules. Let's count from this figure. Okay, this will be more convenient for you, right? I would like to say. So if you'll find here, if you'll see the structure here, now see, let's count start from your red. Okay, so how many red lines you can see? Initially in the first set you can see there are two red line, right? Now we want to incorporate one myosin molecules. Just count one blue. Okay, uh, okay, see, I think there are some network issues, but you just don't need to see what I've written, but rather you must have ever to see what it is there in the screen, right? So just go with that. So we are just need to count the lines. Okay, just you can see the red lines. Okay, so you will find that there are one and two red lines are there. Okay, now further we want to incorporate or cover one blue line. So this blue line is present here. Now in order to cover this blue line we need further two red lines, right? So we need 1, 2, 3 and 4 actin molecules to cover one myosin molecule. Clear? Now if you want to add further myosin molecules, so you need to add one myosin molecules. In order to support that you need to add two myosin molecules. So you will find 4 actin molecules cover one myosin molecule, 6 will cover 2, 8 will cover your 3 as shown in this figure. Similarly, if you find, let's consider there is another sarcomere. Okay, as shown, suppose it's not shown, but suppose you see there are one more pair of sarcomeres. Okay, now you will find that half of the Z line is covered in one sarcomere, so other half will cover another sarcomere, and you need further one half of that another I band. Okay, so that will again comprises that, again you will find the 4 will cover your one myosin molecule, 6 will cover your 2 and 8 will cover your 3, and that way you will find the number of actin molecules required to cover one myosin molecules is always be double as compared to the number of myosin molecules. It will not perfectly double, but yeah, just similar to that. I think now it is clear to all of you that the number of actin and myosin molecules regarding that particular question, if you find it. Now see, if I'll explain you that this question, that the question number 10, it is basically asking about the what will happen if the temperature will increase above 50 degrees Celsius, and what this condition is basically called. Okay, so you will find there are different conditions. So if the amount of heat in the body is increasing, so you'll find you must have aware of albumin, right? How many you all are much aware of omelets. So whenever you are just putting eggs in the boiling pan, you will find it is just coagulating. It's because the protein gets denatured. It's not that they are lost, but they denatured, and the denaturation of protein leads to loss of its functionality. Okay, now the functionality of that molecule get disrupted, so that way the muscle contraction, in order to contract the muscles we need that proteins to get activated. If the protein is blocked, if the protein is denatured, then the muscles is not going to be able to perform its function. Okay, and now this particular condition is occurred because of the muscle protein get coagulated because of the denaturation, and it lost its functionality, and this process is called your heat motif. Why heat motif? Because the motif wood is basically responsible for the loss of its functionality, and it is because of the heat. That's why the process is called your heat motif. Now you will find here there are two other terms that I'm supposed to discuss with you guys. Okay, so you can find what is lever motif then. Lever motif is again it's it's a condition where you will find just instantly after death. Here the persons after death, it's not like that everything is get blocked. Okay, still the blood flow is there, still the movement is there, or the muscles are still contracting because of the few amount of oxygen left behind, few amount of energy left behind with. So this particular condition is called basically pooling of blood or decoloration of blood, is because of the process called rigor mortis. Okay, now if you'll find what rigor mortis is, then now rigor mortis is just after that condition, after rigor mortis condition, you'll find after 2 to 4 hours there is no muscles, there is no energy, there is no oxygen, there is nothing in the body, there is no blood flow, so there is no living organelles in the body, so there is no energy that has been generated, and the muscle contraction gets stiffened, the muscles get stiffened, as there is nothing that can perform its functions for its contractions and relaxations. Okay, so that particular condition is called your rigor mortis. There is one more way rather after this, that this is basically condition that is after 2 to 4 hours of death, and apart from this there is one more condition that is called your rigor mortis. Okay, now what rigor mortis is that? It is basically a condition that's occur when a person gets accidental death. You'll find due after postmortem the body is get cooled for that, and that condition is because of the after the rigor mortis condition. It's not only till rigor mortis, after that it's due to the stiffening of the muscles, the body temperature again going to further cool down, and that condition is called your rigor mortis. Okay, so these are the three terms, that is basically your rigor mortis, rigor mortis, and rigor mortis, along with heat mortis, is that these are basically the loss of functionality of your muscle layers, that is after death, after few hours of death, or because of different post-mortem modification, or it can be because of the heat, or it can be because of any reason. So the malfunctionality of the muscle layer is called basically mortis, and it can be it named basically because of the reason that leads to its dysfunctionality. And that's why the answer of this question will be, when the heat temperature is increasing, the muscle protein gets coagulated, and the process is called your heat mortis. Okay, so this is what about your this particular question is that, if this is what about your muscle contraction and relaxation, is that the muscles are getting uh, stiffened, they are just going to not properly functional because of different reasons, lack of energy, lack of oxygens, etc. Okay, so this is what I was supposed to discuss with you guys for today's classes. Hopefully it will help you guys. Now I would like to request you, if you have any things or from previous classes or like that, or regarding this topic that you like to discuss, just go ahead, so that we can just have a quick look at it. Anybody with any questions or anything like that? Okay guys, so this is the end for today's classes. If you want to discuss anything, or you can just go ahead. Apart from this, you may find this in your YouTube, so you can just go and have a look again. If you are finding any doubts to understand this particular aspects of muscle contraction, just do let me know. And that's it for today's class. So okay, see, I don't think that it is required or convenient for everybody to repeat all the questions again. Rather, you can just go and find the YouTube videos to understand it again. Okay, so this is what and this is basically to improve your concepts regarding all these questions, that how, from where these questions are coming, and how you can just able to better yourself to handle such things. Okay, so it's not like that you need to get the answers, rather you need to understand the concepts, that what it is actually. Okay, okay guys, so I think that's it for today's classes, and hopefully this will help you to understand this particular aspect of biology, and we'll see you in for the next class on next Friday. Till then, all the best and goodbye to all of you. Okay, chalo. Thank you so much for joining the classes, and yeah, thank you all. So that's it. Or anything, if anybody would to like to discuss, just put in the chat box. I'll wait for one more minute. Okay then. Okay guys, then thank you so much. Okay, let's finish it off here.